Hello, my name is John Chatterton. This summer I worked in the lab with Dr. Jim Ravosti. In the lab I studied a mechanism of disease progression in various models of muscular dystrophy. It is known that aberrant production of reactive oxygen species is an important factor in the pathogenesis of Duchenne muscular dystrophy. NADPH oxidase 2, also known as NOX2, is an important producer of reactive oxygen species. Therefore, therapeutic inactivation of NOX2 can serve as a targeted approach to slow disease progression in Duchenne muscular dystrophy.
Hi, my name is Lucas Foss, and I will be a senior at UW-Madison in the fall. This summer I've worked in Dr. Michael Kaiba's lab. My project has focused on the question of how to convert one cell type into another cell type. Specifically, we are interested in converting fibroblasts into blood through the overexpression of certain key genes. Our lab has generated a transcription factor cocktail which overexpresses hematopoietic transcription factors. This cocktail has efficiently converted mouse embryonic fibroblasts into blood. However, adult cells have been resistant to the same changes. As a result, I have been screening various genome-modifying chemicals in order to inhibit this chromatin condensation. It is our hope that these chemicals will allow us to efficiently convert adult fibroblasts into blood.
My name is Matt Jorgensen, and I'll be a senior here at the U next fall. I spent the summer working in Dr. Dawn Lau's lab, studying how estrogen affects skeletal muscles' inflammatory response to injury. Inflammation is necessary for the restoration of form and function following muscular injury, and evidence has shown that in the absence of estrogen, such as in postmenopausal women, this response is impaired. Our goal in the lab for the summer was to use the freeze injury mouse model, immunohistochemical staining, quantitative IHC, and Western blotting to evaluate how estrogen modulates the various aspects of the inflammatory response and hopefully better understand the protection it provides.
I'm Jared Matsky. I'm a senior at the University of Minnesota. I've been working in the laboratory of Dr. David Thomas to understand the mechanism of muscle contraction at a single molecule level. In particular, I've been studying the motor protein myosin 2, which is responsible for converting the chemical energy of ATP into the mechanical work of muscle contraction. This summer I developed a fluorescently tagged myosin 2 and expressed it in the eukaryotic microorganism Dictyostelium. The construct is sensitive to structural changes within the molecule in relation to its biochemical state, and thus serves as a reporter of the structural states that the molecule assumes in live cells. The sensor will be used as a tool for screening new therapeutic treatments of cardiomyopathies.
Hi, my name is Divya Malachuri, and I will be a senior at Woodbury High School in the fall. This summer, I worked in Dr. Michael Kaiba's lab, looking at FSHD, or facioscapulohumeral muscular dystrophy. This muscle disorder is associated with the DUX4 gene. DUX4 has been proven to cause cell toxicity in patients with FSHD. I looked at chromatin modifier genes, or genes that regulate other genes, as well as a set of chemical compounds, to see their effect on DUX4 levels in myoblasts, or cells. Our aim was to understand which chemical compounds and which chromatin modifier genes affect DUX4 expression, to better understand FSHD and hopefully make treatments for patients with FSHD in the future.
My name is Adibella Fojo, and this summer I worked in Dr. Main's lab on a cardiac protein called troponin complex. The troponin complex is a component of the sarcomere that plays a significant role in the regulation of contraction and relaxation of the heart muscle through a calcium-dependent movement. The troponin complex, alterations in the functioning of the troponin complex could lead to or could contribute to heart failure. Heart failure, the sensitivity of the troponin complex is inadequate, leading to poor heart performance. The goal of my project is to carry out NMR studies, also known as nuclear magnetic resonance spectroscopy, on troponin complex to study how non-therapeutic molecules alter the conformation and structure of the troponin complex, so that in the future we could develop more specific and more potent therapeutic drugs to treat heart failure and maximize heart function.
Hi, I'm Katie Reinertz, and I go to University of North Dakota, and I work in Dr. Thomas's lab. I'm working on a drug screen campaign that measures the effects of drugs on the structure and biochemistry of myosin using fluorescence resonance energy transfer. These drug screens will be used to discover novel compounds that will be used to treat heart failure and cardiomyopathy.
Hi, my name is Joel Rosenberg, and I'm going to be a junior at University of Wisconsin-Madison. As a Lillehei summer scholar, I worked in Dr. Dan Gary's lab studying the development of blood vessels with association to ETV2. ETV2 is a gene and transcription factor that is essential for blood vessel formation in the embryo. In my project, I complemented ETV2 mutant embryos with wild-type embryonic stem cells to determine if the phenotype of the blood vessel defect could be rescued, which it was. I also analyzed the way these blood vessels were formed by examining if the cells communicated with each other to send cues to form vessels even without ETV2 expression, but found that this was not the case and that ETV2 acted in a cell-autonomous manner. This experiment gives us insight into how blood vessels are created, and hopefully we can use this to generate vessels that can be transplanted for patient treatment.
Hello, my name is Sammy Shaker, and in the fall, I'll be a sophomore at the University of Minnesota. This summer, I worked in Dr. Albert Tranquillo's lab on a project concerning tissue-engineered heart valves. Tissue-engineered heart valves show the ability to grow and regenerate after implantation to patients, and they can be produced in mass quantities fastly. As such, they're a great target for pediatric patients and other patients who have a long life expectancy after surgery. However, very little analysis has been done on tissue-engineered heart valves in the realm of fluid flow around the valve. And as improper fluid flow can cause thrombogenesis and subsequent morbidity and mortality, this is an extremely important area to look at before these valves are allowed to go into mass production. As such, I analyzed the flow around the valves using MATLAB and TechBot to produce plots concerning the vorticity and shear stress around the valves, and I then compared them to literature example.
My name is Joseph Wenger, and I'm going to be a senior at Cretin-Durham Hall next year. I worked in Mary Gary's lab looking at the role of the extracellular matrix in the regeneration of hearts. Adult hearts are known to lack regenerative ability, but it is also known that for a brief window of time after birth, the neonatal heart of mice possess the ability to regenerate after injury. We were trying to induce the division of adult heart cells similar to that of the neonatal heart cells following injury. This could help us understand why our regenerative abilities are lost and how we could restore them.
Hi, my name is Maida Yang, and I work with Dr. Paul Yazov in the Visible Heart Lab. The University of Minnesota is a unique place that houses a human heart library which contains more than 300 donated human hearts not viable for transplants. Most of these hearts are diseased with hypertension or high blood pressure, which is the #1 killer with heart disease, and 1 out of every 3 adults have hypertension or high blood pressure. My project for this summer is to compare the anatomy of normal and hypertensive human hearts from males and females. The purpose of this project is to determine the variability of hypertension between males and females' hearts. We found that the atrioventricular or the tricuspid and mitral valve diameter increases for male and decreases for female. This finding could help impact the treatment for hypertension.
Hi, my name is Samyukta Melachuri, and I will be attending the University of Pittsburgh this upcoming fall as a freshman. This past summer, I worked in Dr. Daniel Gary's lab, studying the regenerative process of skeletal muscle and myogenic factors involved in this process. Previous studies done by our lab has shown FOXK1, which is a transcription factor, to be expressed in muscle satellite cells during embryonic development, postnatally, and following injury. FOXK1 null mice have growth retardation and impaired skeletal muscle regeneration. My project involved histologically analyzing skeletal muscle tissue injured by cardiotoxin-mediated injury for FOXK1 transgenic mice for FOXK1 expression at various stages post-injury. We also did a series of immunostaining to identify the localization of FOXK1 throughout the regenerative process. Our results showed that FOXK1 is indeed involved in the regenerative process of skeletal muscle, and further experiments revealing the relationship of FOXK1 to myogenic progenitors and satellite stem cells will provide us with insight into the roles of FOXK1 and enhance our understanding of skeletal muscle regeneration.